Hi, I'm Aryan, and today we'll be going over cell division, also known as mitosis. And before we really start to understand how cell division works, we need to be well versed with DNA and how it's packaged. So I talked a little bit about this last time, but in case you forgot, prokaryotic cells like bacteria, which don't have nuclei or other membrane-bound organelles, have a single really large circle of DNA, which we call a chromosome. On the other hand, eukaryotes, like plants and animals, have a nucleus that they keep their DNA in, and they keep it in really tightly wound chromosomes, and these are linear. And the number of chromosomes can vary greatly depending on the organism. For example, most human cells have 46 chromosomes, while fruit flies have only 8, and the current record holder for the most chromosomes is a type of fern called Ophioglossum, pictured here, which has 1,260 chromosomes. So the number of chromosomes an organism has doesn't necessarily correspond to its complexity, as you can see. Now, I said that most human cells have 46 chromosomes. A more accurate statement would be that all human somatic cells have 2 pairs of homologous chromosomes. Now, that's quite a mouthful, so let's really break it down a bit so we can actually understand it. Basically, a somatic cell is any cell in the human body except for reproductive cells, which are the sperm and egg, as you can see. And all somatic cells have 2 copies of the same 23 chromosomes, which are called homologous pairs. This fact that they have these 2 copies makes them diploid, signified by the symbol 2n. On the other hand, germ cells are haploid because they have only 1 instead of 2 of each chromosome. And I showed this in my really oversimplified drawing with these orange and pink chromosomes. As you can see, the diploid somatic cell has 2 of each, while the haploid germ cells only have 1. Alright, just a quick note, the haploid gametes are also known as sex cells because they correspond to the organism's biological sex. Alright, last time I also mentioned that DNA is incredibly compacted in eukaryotic cells. In fact, if you laid out all the DNA in a single human cell end to end, it would be roughly 3 meters long. And if we consider that the human body is made out of trillions of cells, it's really crazy how much DNA we have. So how do we fit all this DNA into a small nucleus? Well, like I've said, the DNA is wrapped really tightly over and over and over again. And let's go over the tiers of this organization. And they're all named after how wide the, I guess you could say, fiber that they form is. Our starting point is the basic DNA double helix, which is 2 nanometers wide. So we call it the 2nm fiber. It's all the way at the top of this slide. And as I went over last lecture, DNA is a polymer of nitrogenous bases. So the first level of organization is pretty straightforward and really well understood. The next level is called the 10 nanometer fiber because DNA is wrapped around these circular proteins in dark blue here that are called histones. And these histones are roughly 10 nanometers wide. The next level are when the histones are then wrapped around into these coils, and these are called the 30 nanometer fibers because that's how wide they are. And then that's at the bottom of the screen here. Next, these 30 nanometer fibers are looped around in a different type, or rather on a different type of structural protein in a way more complex than previously, and that's not really well understood at all, even by researchers. And these then become what are called 300 nanometer fibers. And finally, these 300 nanometer fibers are coiled around into a chromosome, which is about 1400 nanometers wide, as you can see. Okay, so the next thing we're going to take a look at is called the cell cycle, which is basically the life cycle of a cell. Let's start with the most basic part. The cell cycle can be broken down into 2 very different phases, interphase and M phase. Interphase is what the cell spends probably more than 99% of its life in. And in this time, the main things that are happening are that the cell is growing and it's also replicating its DNA. The M phase is really short, and it's when the cell divides. Now, it's called the M phase because the process of cell division for a somatic cell, which remember is any non-reproductive cell, is called mitosis, therefore M. The next few slides are going to be a detailed overview of mitosis, but before I show you all of that, I want to lay down the names of these steps in the correct order. There's prophase, prometaphase, metaphase, anaphase, telophase, and lastly, cytokinesis. Okay, so now let's take a look at the first step of mitosis, which is called prophase. The first thing that happens in prophase is that chromosomes condense. Basically, DNA exists in a less compact form than what I showed you a few slides ago during interphase, and then during M phase, it needs to condense into these discrete chromosomes, which are a lot easier for the cell to organize. The other thing is that the nuclear envelope starts to break down because these chromosomes need to be moved around, and having a membrane in the way could be really cumbersome. Lastly, the centrosomes start to extend out spindle fibers, which are really just microtubules in the direction of the chromosome, or the chromosomes. The next step is prometaphase, which is really just an in-between step that follows prophase and comes right before metaphase. In this phase, chromosomes finish condensing and are definitely visible. Also, the central part of the chromosome, basically where the two lines cross, if you think of it as an X, is called the centromere, and spindle fibers from the centrosome attach to that. I drew the centromere in black here and the spindle fibers in red. Also, by this point, the nuclear envelope is pretty much completely broken down, allowing the chromosomes to move around freely in the cell. Also, the centrosomes move to the opposite sides of the cell. Now, in metaphase, the attachment of spindles to the centrosomes are completed. So the centrosomes the centromeres finally have full control over how the chromosomes move. Because of this, the chromosomes are lined up really nicely on a line called the metaphase plate, which I've drawn here as a light blue dashed line. Next, we have anaphase, which is when each chromosome is split up into two halves, which are called sister chromatids. These sister chromatids both have centromeres attached to the centrosome by spindle fibers, which pull them to the opposite ends of the cell. Also, spindle fibers are used in this step to really elongate the cell. Telophase is when this starts to look more like two normal cells. Basically, the spindle fibers kind of let go of the chromatids they were attached to and kind of disassemble as the centrosome becomes inactive. As this happens, the nuclear envelope begins to reform around each set of chromosomes in the new cell, which we can now call daughter cells. Cytokinesis is the last part of mitosis and is also the simplest. Basically, the two daughter cells officially separate as a membrane forms between them and they both go their own ways. This membrane that comes to separate them is called a cleavage furrow in animals. Okay, now I am going to try to summarize all of what we just went over in the next two slides, starting with the terms. So interphase is basically almost all of a cell's life in which the primary thing it'll do is grow, and then there will be a period of time where it replicates DNA too. Next is mitosis, also known as M phase, which is basically just when the cell divides. Chromosomes are compact structures made of DNA, and chromatids are really just half of a chromosome. Now this is where names can get a little bit tricky, as we have a lot of words that begin with C. The centrosome is the organelle that organizes spindle fibers, and spindle fibers are just structures made of microtubules that move chromosomes and other organelles, and they also help stretch these cells to that elongated shape. Basically, they do all the work to physically remodel the cell so it can divide in two, and so that both the daughter cells can have everything they need. And finally, there are centromeres, which are these locations at the center of the chromosomes that spindle fibers can attach to. Okay, next I'll briefly be summarizing the process of mitosis as a whole. It starts off with prophase, which is where the nucleus dissolves, chromosomes form, and spindle fibers form as well. Then there's prometaphase, which is where the spindle fibers attach to the centromeres after the chromosomes are fully condensed. That's followed by metaphase, which is when the spindle fibers are completely attached to the centromeres, and then the chromosomes are lined up on the metaphase plate. After that, we have anaphase, in which chromosomes are broken up into two sister chromatids, and then those two sister chromatids are brought to opposite ends of the cell by spindle fibers. Also, the spindle fibers elongate the cell a fair amount, and this is followed by telophase, which is characterized by spindle fibers disassembling, the nucleus starting to reform, and chromosomes also disassembling. Finally, cytokinesis is the process of the now two daughter cells splitting apart completely as a cleavage furrow forms. Alright, now let's finish this off with a quick practice problem. Which of the following cell components are involved in that are involved in mitosis are made of microtubules? Only one of these answers are correct, so I will give you a few seconds. You can pause the video, try to figure it out, and then I will go through all of them. Alright, now let's go through these answer choices. First of all, the nucleus definitely isn't made of microtubules, so that's not a correct answer. Chromosomes are made of DNA, and chromatids are just half of a chromosome, so they are also made of DNA, and this means that both those answers are wrong, and the mitochondria isn't really even involved in mitosis, and even if they were, they are not made of microtubules. This leaves us with spindle fibers, which are in fact structures made of microtubules. And that about wraps it up for the lecture on mitosis. See you all next time.